Who are we? So Mesoblast is a regenerative medicine company founded in 2004. There were a number of investigators at Columbia University who were interested in taking stem cell therapy into the clinic, and we, including myself, were feeling a bit frustrated about funding mechanisms and time to get into clinic with the current status in academic medicine. And so our founder, Silviu Itescu, licensed a technology that we thought was very promising for immunoselecting and culture expanding mesenchymal precursor cell from the bone marrow. And then we subsequently developed that cell, first by working in some preclinical models, to promptly get into the clinic to test some of our assumptions about mesenchymal cell therapy for tissue repair and other regenerative medicine indications. Just on this slide is just a snapshot of where we are with the company now. We are publicly funded. We're traded on the Australian Stock Exchange, and we have a history of having had some successful fundraising experiences with the Australian and also UK investors. We also, in 2010, signed on an alliance deal with a commercialization partner, which was Cephalon in Fraser, Pennsylvania, and began to work in partnership with Cephalon on commercialization on 3 of our programs, the most advanced of which is cardiovascular medicine. And then subsequently, Cephalon was acquired by Teva, so we've undergone a transition, an adjustment with a new partner, and are continuing to move forward with commercialization of our programs with Teva. So just to talk about where we are with programs now, as many of you know, we're in a number of areas of development, and so even though it started out with programs in heart failure and in bone repair, we've since extended into a number of additional indications. So in terms of our recent key highlights, we've established long-term follow-up for the first 60 patients in a phase 2 trial of congestive heart failure, identifying the MPC dose, which prevented any episodes of heart failure, hospitalization, or death out to 30 months. And so together with Teva, we have worked with regulatory agencies to discuss the proposed phase 3 trial design, which we expect to execute shortly. We've also initiated a phase 2 trial to prevent heart failure after a major heart attack, and together with Teva, we are opening that study across sites in Europe and Australia. And we've just recently completed enrollment in phase 2 spinal fusion trials, and a related indication, which is degenerative disc disease, will be completing enrollment in the next month. Additionally, we've recently embarked on a new franchise, which we call the Intravenous franchise. So to date, all the other indications you saw on the prior slide were direct targeted delivery of cells. And so this program is actually looking at systemic intravenous delivery of cells for a number of indications. This based on some preclinical findings which are positive in a non-human primate model of type 2 diabetes, which supported commencing a phase 2 trial in type 2 diabetes for glycemic control, and then subsequent development to look at some complications of diabetes. And at the same time, we were working on a model of inflammatory arthritis in sheep, and in this large animal model demonstrated that there were multiple pathways that, inflammatory pathways in rheumatoid arthritis that seem to be affected by mesenchymal cells. And so based on the positive findings in that model, we are working now to initiate our phase 2 program in rheumatoid arthritis. And we've also had to keep in mind what the potential challenges are for manufacturing, and so we've implemented a number of product manufacturing strategies to ensure that we have a commercial scale-up available for all of our programs, so that we can also reduce our cost and assure that we have capacity for commercial supply for our products going forward for many years. In terms of what our business model is, you know, my disclaimer already is that I'm not a business development person. So we have a proprietary platform technology. It delivers multiple product pipeline. They're what we call multiple shots on goal, and we're not really dependent on the success of any single product. So as I mentioned earlier, we're in heart failure with a intramyocardial delivery system, but we're also at the same time developing the IV franchise, which is targeted towards inflammatory disorder, and so that's a totally separate patient population and a separate method of delivery, but based on the same platform product. We have successfully established corporate partnerships to help manage our execution risk. We work closely with Cephalon and Alteva to provide the global distribution capabilities and the regulatory and clinical trial expertise for commercialization that they provide to us. And we also have worked with a commercial contract manufacturing organization, Lonza, which provides for us manufacturing expertise as well as global process development and capabilities. We've also established access to a Singapore facility which alleviates our needs for internal spend on manufacturing. So I'll describe this a little more on a subsequent slide, but we've worked closely with Lonza to map out our commercial manufacturing needs for the short and the long term. We're fortunate to continue to maintain a strong cash position, which is enabling us to simultaneously develop multiple product areas. And so Mesoblast has sufficient cash to advance many new programs in parallel. And we have also been fortunate enough to recruit and maintain a strong core group of people with expertise in cellular therapy in areas very relevant for our programs. So this is part of the general de-risking strategy. And just a little more discussion about our relationship with Lonza. As many of you know, Lonza is a very experienced contract manufacturing organization for cell therapy, and our agreement with the Lonza is that they will supply both the clinical short and long-term commercial products to meet our needs globally. They are constructing a purpose-built manufacturing facility that will be for our exclusive use that at some point we may or may not buy out. Mesoblast will have exclusive access to the cell therapy facility being built in Singapore. But although we are utilizing that manufacturing expertise, we do maintain control over the manufacture procedures and goals for all products. And so we work on establishing how we will delineate our products and for different indications, and work very hard to control our costs and obtain optimal pricing for manufacturing and also for the goods for different indications. And so the commercial benefits of this arrangement are overall reducing the cost of goods, hopefully increasing our margins further on sales price, and utilizing the state-of-the-art industrialized manufacturing procedures for both R&D support and for commercial development. So just a bit about our technology. We are a bone marrow-derived mesenchymal cell that is isolated by immunoselection. The end product is a potent, purified adult-derived cell, which has a strong safety profile and certainly avoids many of the ethical and safety issues that were raised in years past about other stem cell sources. We're backed by a strong patent position. We are a product that's off the shelf, so once this product is harvested from the bone marrow donor, immunoselected, and cryopreserved, it is available off the shelf for use. It does not require matching for HLA or ABO or any other type of tissue matching for use, so it's a universal donor type strategy. We feel we've had very clear and fairly well clarified regulatory pathways in the regions that we've gone to date. The cells can be easily expanded to large numbers, so we have fairly low cost of goods and no supply constraint, and so the model is one of a high margin. And just to describe a little bit more about how we get the cells and our IP position is that we own IP on composition of matter, the manufacturing product as well, the process as well as the global use, and so it covers the immunoselection, the expansion process, and these and a number of other indications that we are exploring. And the areas that we're involved in now include, as I mentioned earlier, a number of different indications. There's the IV program franchise, where intravenous products are being explored now in type 2 early diabetes. So we're also shortly embarking on these additional indications. As I mentioned, the cardiovascular program, we're also working in orthopedics and in eye disease. And this is our product pipeline, so we are in some areas in phase 2 and promptly moving into phase 3 in some other indications. And this is just a little bit of background on where we are with the cardiovascular program in heart failure and sort of how we got to where we are at this point. We've conducted a phase 2 trial, which has been the results of which have been announced publicly. And with Teva, we've jointly attended phase 2 meetings with major regulatory bodies to assure that we have alignment and understanding of their expectations for endpoints for study conduct and other issues that will help us to successfully complete this phase 3 study. This was based on the phase 2 study results, which showed that identified for us that the highest MPC dose completely prevented any episodes of heart failure, hospitalizations, and death in a follow-up period, which is now extended out to 30 months, and that was compared with a 25% event rate in the control subjects. And the highest dose also showed us evidence of remodeling by reduction in cardiac volumes and also improvement in functional capacity by the improvement in the walk distance in the cell treated compared to the control patients. And so the proposed phase 3 trial for product approval has been powered based on this event-driven efficacy endpoint, which we were fortunate enough to be able to demonstrate in the phase 2 program. And this is just the visual depiction of that, showing the what we call heart failure event-free survival for the cell-treated dose, the dose that performed the best, 150 million, compared to the control population. And as I also mentioned, we are working forward, promptly moving forward, independent of our alliance with the IV franchise in the indications listed here. And this is an IV formulation of the allogeneic cells, again, which is targeting a number of systemic diseases. They address large global markets and represent significant partnering opportunities. In diabetes and metabolic diseases, we're already in our phase 2 looking at glycemic control in this population and establishing safety and tolerance of the IV preparation. And in inflammatory and immune diseases, we are about to convene our phase 2 in rheumatoid arthritis. The background, a little bit of background just on 2 of these programs is for diabetes. As we all know, it's a huge market opportunity with approximately 19 million Americans are affected by type 2 diabetes. And our IND-enabling preclinical studies demonstrated that we were able to show early and sustained reductions of glucose by a single IV injection of allogeneic cells at 0.31 or 2 million cells per kilogram. We also observed reductions in CRP levels, suggesting a potential cardioprotective effect of the MPCs, which for folks involved in clinical development of diabetes interventions, that demonstrating a cardioprotective effect in diabetes development is important point to stress, in that many hypoglycemia-inducing agents have actually established cardiac risk. So part of the development requires our establishing that we are not inducing cardiac risk with our product. We also, in this preclinical program, demonstrated that repeat dosing was safe, and that early onset glucose-lowering effects were demonstrated also along with physiologic responses in osteocalcin. And this just highlights some of the sort of the non-conventional thinking that's taking place in our preclinical development to try to understand how mesenchymal cells exert their effects. And we are probably very close to the opposite of many of the large pharmaceutical organizations, where a complete understanding of the agent takes place before there's ever entry into the clinic. And so this is an area where we've worked to try to understand why this could be relevant in diabetes based on what we've seen in the literature, what we've observed in our preclinical studies, and really to understand what are the systemic etiologies of this disease, and so why would we take an approach to systemic therapy as opposed to some other interventions being investigated in diabetes. And we also, in a type 1 model, streptozotocin rodent model, demonstrated the increase in exogenous insulin production following mesenchymal cell therapy. So the diabetes clinical development program looks like this at this point. This is an ongoing randomized placebo-controlled phase 2 study, 60 subjects with type 2. The endpoints are establishing safety over 3 months, and glucose control is a secondary endpoint. And our development plans are to expand this therapeutic program to actually look at the unmet needs, complications related to diabetes and metabolic diseases. And so we're certainly looking now at renal complications and also some other organ-specific toxicities in metabolic disease. And in RA, also this is a condition that affects a large proportion of the American and worldwide population. It's estimated to be about a $10 billion global market, which continues to grow and afflicts about 1.3 million Americans. And a diagnosis of this disorder is associated with a shortened lifespan and an increased risk of cardiovascular death. The incidence of RA diagnosis increases with age. And there's a clear clinical need for novel single-use remission-inducing therapies. The agents that are currently available are useful, but do not induce remissions at a high and consistent rate. And so the theory is that IV MPCs may affect multiple disease pathways simultaneously to exert an effect on rheumatoid arthritis. In the preclinical setting, a collagen-induced arthritis model established established model for human RA showed that a single IV injection migrated to the inflamed joints and the lymph nodes, and this significantly reduced synovial tissue levels of the cytokines noted and inflammatory cell infiltration. And it also significantly reduced the histopathology findings that were measured as, oops, I hope I didn't do that, in this model. And there was a dose-dependent efficacy demonstrated. And so the clinical development program for RA looks like this at this point. We're planning a randomized placebo-controlled phase 2 study. It'll be a single infusion of 1 or 2 million cells. And this is just the criteria for entry. And at least as early as 3 months, we're going to look for responses and by established criteria, clinical responses, but certainly expecting to follow out responses, particularly radiologic evidence of joint damage all the way out to 12 months. And just in closing, I just want to just describe what our upcoming inflection points are for the company. The continuous focus on the IV franchise with the programs I've just described, completion of the phase 2 spinal fusion trials, evaluating, analyzing data, making decisions about these, completion of our phase 2 degenerative disc program, and then commencing the phase 3 for heart failures. And we continue to look at all of these as opportunities for additional opportunities. Obviously, we're very comfortable and confident that we can continue to develop these programs, and that really allows us sort of the luxury of waiting for partnering opportunities that are really of most benefit to us and the potential partners. And so with that, I will close. I think I'm just about on time.
Yeah, so we're going to move on because we are the last 2 people standing between these people on lunch. So I think we'll table questions, and if people want to track Donna down afterwards, you can find her in the outside. So I want to thank Donna.
Okay, thank you.